Howdy, folks, and welcome to Tech on Drugs. With me today is Master Drug Developer Tom Bumol. Welcome, Tom.
Thank you.
Tom is a Senior Vice President at Eli Lilly, where he was headed the biotechnology and immunology arm and was also the president of the San Diego site. With his colleagues, he developed over 100 different molecules, 90 of which are in various clinical trials, and 11 of which have already been approved, some of the leading assets in the industry. He then moved on from pharma to the Allen Institute, where he was an executive vice president at the Allen Institute and the founding director of the Allen Institute of Immunology, paying special attention on studying human immune variation in health and disease. We'll talk more about it and its importance. And then since 2022, Tom has decided to focus his energy with companies and students and really kind of cherry picking the right companies to be working with to actually make an impact on changing how drug development is done. We at CytoReason are lucky to have him as part of our scientific advisory board. So Tom, real pleasure to have you here.
Thanks again.
Wonderful to have you. I mean, in fact, I think your record is going to be, can really shed a light on the stages drug development is in at right now, which, thinking about kind of moving from trial and error to predictability, from monotherapies to combinations. We'll be touching on all of those topics today. But to start us off, just maybe a few kind of quick questions we ask every guest here in this podcast. And just first would be really what you think, from your perspective, is the industry's biggest achievement that you've seen in your career.
Yeah, I think perhaps the biggest achievement that has occurred is the rise of biological therapies of the last 20, 30 years, whether it's monoclonal antibodies or therapeutic proteins, but adding that arm of opportunities pharmacologically with designable specificity and other things have just really been impressive and has changed the landscape of several major human diseases. So combining that with traditional small molecules, biologics, and now for genetic medicines, at least we have a lot more tools in the toolbox to tackle complex human diseases. Cool.
And from the flip side of the coin, where do you feel the industry has failed the most?
I think, as I'm sure many have stated with you in the past, this is basically a failure business. We hire the best scientists in the world, and yet 8 out of 10, 9 out of 10 times, the concepts that look great preclinically and early R&D often fail. And so what is the basis of that failure? Is it the technology? Is it the disease? Is it the humans we're trying to treat? These are all really important questions to understand, because with today's technology, we can ask deeper questions about that complexity and begin to sort of unravel it and hopefully find a path to improve the probability of technical success.
Yeah. So for the mice in the audience, I got wonderful news type of.
Yes. No, well, I mean, the joke that people have often used is humans are not inbred mouse models, and we are wonderfully heterogeneous, and that heterogeneity has to be embraced in drug discovery and development, and whether it's any therapeutic area, including the human immune system. And the human immune system, is a wonderful example where we can show easily how incredibly diverse that is, particularly with the epigenetic phenomena that can affect our fundamental immune health and immune dysfunction.
Sure. So yeah, heterogeneity in disease biology as a failure, we'll talk a lot about that today. And on a more personal level, where do you see your kind of biggest success to date?
Yeah, well, I was fortunate enough to be part of a merger and acquisition of a company at Lilly that was a protein engineering company. And what was exciting to me about that was that we not only increased our capabilities of more candidates, more shots on goal, but we also increased the design, both for efficacy and druggability and safety, dramatically. And because of that, we began to see some really exciting drugs that emerged that, quite honestly, surprised us and continue to surprise us, being highly efficacious. It really is the difference between target-specific therapies versus broad pharmacology. They're both powerful, but I think we've now seen the emergence in the last 15, 20 years of targeted therapies and how important they can be.
So many of the successes, I guess, of those clinical trials, those drugs that you've made into the clinic really came from that initial engineering approach and ability to build specificity into it.
So a good example for us would be the early days of IL-17A targeting. Lilly has on the market a drug called ixekizumab, which targets IL-17A. But this is a great example where the specific targeting, but then also the context. If it was run routinely in all autoimmune disease, it would not look as potently active as it is in psoriasis. So it wasn't just building the drug, it was also understanding the medical condition in which that system could be perturbed to give the best outcome. And I think the surprise for me and the rest of the world then was one of the first times in my career after 30 years at Lilly was that we actually doubled standard of care efficacy in psoriasis. Yeah, when you see that, you don't forget that. That's one of those things I'm saying, okay, that's not a common phenomenon.
Made your day.
Yeah, made our day, made our year, something to remember. But I think, so the real important challenge for the future is, how do you repeat that?
Right.
How do you think through the parameters that allow you to see such powerful medicines happen versus incrementalism? And I think, to me, that is the opportunity that the new technologies wed to R&D could really help.
No, that's really in many ways what we're going to be talking about today with predictability, trial and error to predictability. Maybe just before we get going with that, in this long-spanning career, I'm sure there's many achievements and ups, but also some downs. So if you have to pick like a story of failures that you've experienced that you feel like there's something that we learned from.
No, it's a great question. None of us like to talk about our failures very much and missteps, but one prominent one, my colleagues and I at Lilly pursued the candidate drug called tabalumab, which targeted BAFF, B-A-F-F, and we wanted and definitely believed that we had a superior asset, and we went into lupus, SLE, and we failed. One trial had a partial active signature, and the other trial failed. And these were 800 plus patient trials per group, very expensive, very long term, many people involved. Going back and looking at that data was an eye-opener for us, because we realized what was characterized as lupus actually was many diseases. And even using simple signatures, such as interferon, such as complement levels, topics like this, some of this is published by my colleagues, we could actually see that the drug would work in certain populations, but didn't work in others. Yet, in an all-comer trial, we were treating everyone, and hence the efficacy got diluted by non-responders. So this happens all the time. Everyone goes back and re-looks at why a drug worked, and the lesson learned there is disease heterogeneity is real. What you believe is the disease is incomplete knowledge of what the disease is. You have to embrace that and begin to think about it. So actually, that work and other work similar to that actually stimulated me to go to the Allen Institute for Immunology, because there we had the opportunity to start investing in core technologies that can actually help us look at some of the inherent heterogeneity in health and disease.
Well, a failure and a lesson learned, I guess. So
Absolutely.
So moving, I guess, maybe to the main kind of discussion, I wanted to really kind of get your insight on is your story right now exactly captures this idea of there's a trial and error type approach versus trying to get to, you mentioned predictability, non-incremental, but rather a jump. and I guess this is where we all want to go. Getting there is not as easy as we imagined. And so maybe the first question is just to ask, where do you think are the kind of large hurdles with this, and where the investments need to be made?
So I think you have to split it into two ways of thinking. First, the design principles associated with the drug, the intervention. I think we have to incorporate a lot more data about the target disease and the MOA. And to really understand mechanisms of action, you really have to understand the system. And most of us were trained as reductionists. And we like a target, we like an inhibitor. But as someone like yourself who's a system biologist, we obviously know that's inadequate. So toxicologists and pharmacologists tell us more about a drug, but the designers need to understand this, that there are many other things you have to understand about designing a drug. So that's the first part. The second part is, what's your target disease? And where are these parameters best present so you can enrich the population for an intervention? If the goal is therapeutic index, that's number one, safe, effective drugs, but number two is the right population. So I think we're going to have to start asking questions about more precision approaches, about subjecting our populations to subpopulations.
Do you think so? That type of approach, obviously, I sing to in the same choir, and yet what I believe has been a resistance in the industry in many ways, or I'd love to hear your thoughts. Is that that type of an approach does suggest that ultimately sales per product per asset are going to drop because there's a smaller population, possibly, and I think that's maybe where things are missing. Success rate would grow up, right? Yes. And I guess some of your thoughts on that battle on the business versus the science side of this, and how you see it kind of advancing.
Yeah, no, it's the absolute key point to dig deeper into. I think we've, at least I've grown up in a biotech, biopharma world where the strategy was one size fits all. And, the disease gets diagnosed, you treat with drug X, you get Y outcome. A careful look at even the most successful drugs on the market suggests they only work in a subpopulation. So without naming names, everyone could do their homework. I mean, this has become pretty obvious. And so I think what we have to do is embrace that. And another way of thinking about it is to ensure success. Saying if you have this characteristic and you get success, people will take the drug. And with not saying necessarily guarantee, but a path to a greater outcome. Some of the diseases are big enough to separate and get great outcomes, but others will maybe have to think about combining efficacy modalities to increase the percentage of response in a population, which is a separate topic perhaps, right. But the idea is that if mechanism of action 1 works in a population and a little bit in population 2, if you add mechanism of action 2, could you get some synergy there between that? I think that's probably where the big opportunity is going to be in the next 10 to 15 years, is what can we mix and match by design to actually get a broader swath of the disease population to respond to a therapy.
So basically, combination therapy is a means to an end of precision medicine, not in that you're narrowing down a population, but rather you've built an asset that covers all.
Well, it's a compromise to make sure my sales and marketing colleagues appreciate that we are thinking about helping as many patients as possible.
Of course.
And so that would be the idea. But yes, that is essentially it. I think it's time to consider that seriously. And I think oncology is starting, but now with many routine chronic diseases also being subject to biopsies and to more analysis, with the new technologies evolving now, with multi-genomic, proteomic efforts, even the microbiome, I think we have an opportunity to really think about this in a very different way going forward. All these technologies are robust, they're exciting. I had a chance to investigate them for 4.5 years at the Allen Institute. They need to be employed. And employing them and gathering the information in the public domain to create more data, more knowledge about human disease is one thing, but then applying it specifically to development and drug discovery will be great.
So in many ways, I'm thinking now of what you're saying is, combinations as a means of precision medicine. And it sounds to me like in many ways, it's also a, circumventing to some extent the need for truly understanding, fully disease biology. Because I'm covering multiple bets, right? Together.
No. I mean, it's one way of looking at it, but I think the natural progression. We used to always have this sort of general concept back at my old company that there was always a first-in-class kind of drug, and then there was, the best-in-class drug, right, which was way better for a variety of reasons: patient convenience, efficacy, all the things you can imagine, less safety risk. And then finally, what was next? So if you look at the evolution of innovation in sort of that mindset of first, best, next, where do you want to fit with your innovation from your company to succeed? Everyone believes at the beginning they have the best shot on goal, and
they believe they have the best even though it's the first.
Yes. and I think this is the challenge. I mean, embracing all the complexity, all the novel pharmacology, and understanding not only that, the history of disease, not just disease heterogeneity, but all the other drugs patients got, which change people, patients. And so integrating those pieces, integrative physiology to design better trials could work. If we're smart enough, and I think the future we will be smarter, I think you'll have an opportunity to logically pick combinations. Instead of just hoping it will work.
Right. No. I did not mean to say that you're when we think about combinations, we shouldn't be thinking logically. I guess my point was that if I am moving into combinations, multi-specifics, and so forth, I can, I guess cover multiple bets. Even as you said, some populations, only have a certain, just a bit of that, say part of the disease architecture that is affected. So
yeah, but I think it just gives you the opportunity to do more method studies and more assessment of what you'd like to do, because I think everybody wants to participate in this. I mean, from an academic perspective, this is new knowledge. Yes. This is new disease biology. From a pharma R&D perspective, this is improving the problem of probability of technical success. Correct. So why couldn't there be stronger marriages there between those two interests and applying it directly to translational research?
Yeah. It also speaks, we were talking about subpopulations before, but, I like to think about, I guess, precision medicine as a means to an end. As in, it's not about taking the subpopulation. Ultimately, it's about studying disease biology and heterogeneity at a resolution of the subpopulations, and then allowing you to, as you were saying, logically pick targets.
And again, I think when you look at the world today, I mean, we are seeing more evolution of approaches. I mean, the whole immuno-oncology side is a great example. Where these drugs not only generated Nobel Prizes, but they also generated great excitement about providing survival advantage to patients that previously had not. But even there, 20, 30%, maybe 35% of the patient have long-term survival. It leaves a lot of opportunity on the table.
Sure.
If you look at the last 10 to 15 years, there's been many, many shots on goal, many combinations, many bolt-ons. But we haven't yet quite achieved that second step function.
Right. I was going to ask about that. Your thoughts on I guess, why is that happening? Why is there? It's almost like a punctuated equilibrium. Right? It was like these step functions, and then very small changes.
No, it's I'm a cancer survivor myself, so I think about this personally. What happens if you're an inadequate responder, and everyone wants to understand what are the next options. When you have this step function in efficacy, sometimes referred to the IO drugs, sometimes referred to as the cisplatin moment of our generation, how do you improve on that? And believe me, there have been great combinations and opportunities to improve it. But what's remaining? And I think then, not to belittle these great, outstanding efforts, good shots on goal, but the simplest idea would be to take what's in my toolbox and throw it on top of it. Because particularly in cancer, combination therapies have oftentimes been done empirically. Adding this, adding that, subtracting that, getting a better outcome. So there is some logic to the design of combinations and combining things. What if you could do that by design principles based on the target disease, instead of just taking what you have and adding it, actually designing things that are going to hit the populations you want? That's at least the idealistic future.
So in many ways, you're saying a lot of the failures that we're seeing in getting, combinations to work, and in any achievement where we have suddenly a step function is just maybe a rush without extensive amount of investigation, if you like.
I'm sure this will ruffle a few people's feathers, but I think that is basically, there is a mentality that you have to get out there and see if your hypothesis is correct. And sometimes you're opportunistic and you go quickly. But a certain percentage of the trials probably could have been thought through a little bit more. In my opinion.
Maybe even going back to basic principles and basic research of understanding what remains once that step function happens.
And then the other fundamental strategy, even in chronic diseases, that perhaps a little different strategy is when you're treating people chronically and you get a level of efficacy. Well, the logical thought is, we'll add another drug and get the level of efficacy doubled, or at least up 30-40% instead of incrementalism. Well, oftentimes the combination of two pharmacologies gets you greater safety risks. Yes. So how do you circumnavigate that? And this has been seen. People have seen, particularly in immune modulators, increased infection risk, loss of tumor surveillance strategies, things like that, which are important not to have as well. So I think that's going to force, and it is forcing, I think, the design of what we'll call conditionally active therapeutics, where you're taking advantage of some fundamental mechanism that's proven, but you're adding an engineered aspect that will bring something that, if given systemically, could be toxic, but given in this conditional construct, only works at the disease microenvironment. That, to me, I think, is an exciting possibility. If that can be developed, we will suddenly take all this great disease biology from biopsies and other things and begin to say, how do you design a drug for that situation? Right.
So basically, you'd drop the safety risk in that end and get the added value of the combination from the disease architecture perspective.
Absolutely. We're seeing the advantages now of bringing more toxicity, toxic drugs like from antibody drug conjugates, ADCs, radiopharmaceutical conjugates, bringing radiation to the site instead of systemic. These are good examples of doing this. Well, now we're talking about biologic combinations that could be doing this. And I think that's going to be really, really outstanding and an opportunity.
I totally agree and excited, but I think there is a big difference between when I look at the ADC space where even there, the payload we don't fully understand often, right? But at least, I kind of know it's a relatively simplest thing I'm bringing in when I'm talking about these two biologics, bringing two cells together. It becomes even, we don't understand that yet.
Oh, it's an exciting, I completely agree. If you talk about cis and trans biologics, you can bring a cell together with another cell, you can bring two components in a cellular system together or antagonize one, agonize one. There's just a large palette of opportunities for design. But the designers need a blueprint. They need a systems blueprint. And I think that's, and this is where reductionists need to embrace systems and understand the ups and downs that occur when you modulate systems.
So again, I fully agree, and yet, having as an industry veteran, I'll ask you, what do you think are the hurdles to do that? At multiple levels of an organization or a thought of an asset, right? So ultimately, you need decision makers to address, to embrace that type of thinking.
Yes.
As you were kind of saying, both within a disease, for choosing a target, for choosing subpopulation, but also thinking broadly about diseases.
Yes.
Non-trivial.
It's not trivial, and it will be slow. I mean, you would have imagined, if you go back to, when the Human Genome Project was completed and people were talking about precision medicines and everything else, we're decades later, and very little. We're still talking. So the question is, who's going to embrace it? And I think when you see a true proof of concept that generates a big hit where people make this work, the industry does seem to follow success a little bit lemming-like. Why? So I think we're still waiting for that example. And I think some are brewing, and I think people are going to do it. But you're right, it's much easier to think about the disease as an entity, treat it, be successful in treating, with a 30, 40% outcome and, celebrate. Believe me, it's so hard to get there that I understand why it's celebratory. Yeah. But on the other hand, if you're really thinking about the future, you have to really think about, well, how am I going to design medicines that are different, even greater disease-modifying agents than the ones we have today?
Sure. Do you think that's ultimately the industry is going to need to be forced to think there because of payers? That they were going to basically say, look, as you were saying, we look at the stats. Not everybody responds to all the drugs.
Yes. No, it's a great question. I don't pretend to be an expert with payers, but one of the rationales for multi-specific biologics is 2 or 3 for the price of 1. I mean, literally, one could start to think about it that way, which may be scientifically based first, but second, could actually avoid multiple drugs being taken, each with its separate own price tag. And I think that time is coming. And I think we're going to start to see that evolving. But make no mistake, you're absolutely right. The industry today is just learning and seeing what the opportunity is here. But it's coming. I mean, the more we see explained why drugs didn't work versus working, I think there will slowly be, first in maybe mid-sized companies and some future-thinking large companies, to embrace this hand in hand at design principles, as I mentioned before, and in first POCs, phase 1, phase 2.
Right. Do you see the industry? You kind of mentioned this indirectly, but there's a very strong kind of underlying tone of study or failures. Bring those failures that you took all the way to phase 3 to the beginning of your next, your next design. Do you think this is something that the industry has embraced in studying the human immune variation, or kind of the human variation in the disease, and from those principles and bringing that?
I think they should be doing more. Would be my first comment, but they are wondering why large phase 2s and large phase 3s have failed when all the work leading up to that, preclinically and clinically, suggested success. After action reviews have to happen. We have to look at these studies where millions of dollars have been spent. And I would, make the plug that, take samples from these trials, bank them, do other things, prepare for this, because in your lessons learned in an after action review, you can go back. Right. And you can ask the questions with the human metadata and the samples you have as to why things didn't work or worked. That's going to not only open up, whoops, we missed that, or we didn't, we did something we shouldn't have done. It's going to open up new opportunities. Mm-hmm. I mean, I think personally, doing this is also going to open up the opportunities for second and third generation agents, because you'll see the complexities inherent in the disease, such as lupus or other diseases, but then have the opportunity to say, hey, that's an obvious bolt-on. We should try this bolt-on, because look, in humans, not preclinical models, we are seeing this signature persist. Mm-hmm. and that signature needs to change. So I'm. You can argue that I have decided to drink that Kool-Aid, although we're just drinking water. But I think the idea is got to happen because I've had the chance to see both worlds. 3.5 decades in large pharma, but now, thanks to the Allen Institute, the opportunity to see the power of these deep profiling technologies, and more importantly, both vertical and longitudinal integration, which I know you're a big fan of, because this to me is going to tell the real story of how patients are responding to the drugs.
Right. So I couldn't agree more. I think getting those measurements done, getting the industry to embrace this is still something that seems to be not in everybody's minds, and bringing that data to early stage next time is a non-trivial thing, right? And in fact, especially if you start thinking about not just the asset that you just pushed through and possibly, unfortunately failed, but the fact that you need to be thinking broadly across all diseases. How do you get, a decision maker who, goes to sleep and, in the morning needs to decide that we're going after this particular clinical trial? How do you get all the data there so that to be informed there? And that becomes even bigger than any one company's trials.
Yes. Well, as I talked to some pharma companies when I was at the Allen Institute, I got this argument all the time, and it was usually framed in cost.
Cost of profiling.
Cost of profiling. So my plea to the audience here, particularly molecular biologist aficionados, is to democratize this, get this into simple ways to do profiling that would actually help support multi-site clinical trials, global clinical trials, where people can learn and gather the information, because some of the tools we used just had demands that were unrealistic. You have to have an RNA sample less than 4 hours old, preserved. You have to have this and this and this. And when you're running the per patient site for big trials is so expensive, it's logistically impossible.
Right. They work in an academic lab environment, not in the real world.
Exactly. So I think there could be technological improvements. I know some colleagues at the Allen Institute are thinking about this, which could really help, because democratizing it, making it cheaper, making it available, you can do it anywhere, is going to add to the data. And that kind of data is going to help future decision making as well. Plus provide this background information on the successes and failures.
So solving in many ways the boring problems of logistics, integration, and so forth is actually going to be what is going to push us and catapult us forward.
I think there's technological answers evolving, and let's hope they evolve quickly.
Yeah. Another direction I was going to ask you about combinations, as we're seeing this shift from switching topics a bit, but seeing this shift from oncology as being the place people are thinking combinations to immunology, chronic diseases, and so forth.
Yes.
As you've seen this and kind of seen the successes and failures, as you mentioned, in oncology, what do you think people thinking about the immune space need to be thinking of? Because there are no successes really there yet, right, in many ways. And also that's true of biomarkers, right, which in oncology, at least on the I guess genetic side, there's already in particular cell populations there are successes.
No, I mean this is again, as a cancer survivor, I wish there was more progress. I've met scores of patients in my cancer journey who were looking for answers, and the patients are out there, and so you want that to happen, and we're all going to be patients. We all know patients. If you make it personal, we should all work to make that happen. That said, I'm concerned. I'm concerned that we have to sort of turn it on its head and ask what was the reason why we haven't seen more step function in efficacy. And I think part of it is, we'll treat cancer as a chronic disease, so there'll be treatment 1, treatment 2, treatment 3, treatment 4, treatment 5. And look, we've really improved life. Believe me, that is to be applauded. I mean, there are many diseases. Multiple myeloma is a good example. We study at the Allen Institute where survival is much greater now. Lymphoma, much greater. But in many cases, some of them are not cured. And so the question is, what how do you treat from this current paradigm, which is let's add first line, second line, third line, fourth line therapies to get better first line, better second line, where we can actually put the diseases in remission. Cancer, you get away with a more narrow therapeutic index, so I think that's possible. And now you see the emergence of biologics and even biological therapies, cellular therapies earlier and earlier. Mm-hmm. I think that's going to be interesting to see how that plays out. I think it will potentially show that if you hit the cancer earlier and earlier with these therapies, it will help. Chronic disease is a different question because you have to maintain safety. You have people who might have an autoimmune disease for 30, 40, 50, 60 years. You have to be very careful about immune suppression. So you have to target and pinpoint immune suppression. And it's not been that easy to add drugs together with other drugs because...
To add in terms of, not eliminate or kind of generate a greater immune suppression than one
Yes,
wants.
Because of what we talked about earlier. It's just infection risk and other things that occurs. But I would say in the last 10 years, there have been a lot of exciting findings. One my group participated a bit in was the immune checkpoint agonists. That was not a rational thought when it evolved, but the idea that if there was a natural braking system for the immune system that exists when the immune system goes up and comes down, could we power break that? Right. And so a lot of companies are pursuing that strategy. That is very novel. That's very orthogonal. And I think that's going to add to the piece of, can you have safe, natural immune resolution therapeutics to combine with activation phase therapeutics? That's exciting.
Right, and also I guess the conditional component here is
yes, so then
key.
If you have something you know will work, but you can't give it, I mean, the whole idea of targeting something for conditional disease microenvironment immunosuppression is an exciting idea. Whether it's the gut, the skin, whatever the situation may be, that's going to be possible. And if those work, I think that's going to open up a brand new set of opportunities. As we said earlier in the podcast, we'll want to look at where drugs failed and what popped up and begin thinking deliberately on design principles going forward.
Sure, that leads me actually to the technological angle. If I think about how would AI kind of come into this idea of thinking about combination therapies? To me, it ultimately, the way I guess envision this in my mind, I see like a disease landscape. And there's many such diseases, and I can break it down to subpopulations, and I need to ultimately understand perturbations on this on a multi-scale level. Yes. And I guess I'd love to hear your thoughts on
No, I think I'm probably going to give the answer you're expecting me to give, but if I think there's something important going forward, we need to take advantage of the history of pharmacology and disease X and map it accurately, not just competitive landscapes, the successes, the failures, the surprises, and then that needs to meet the sampling and disease databases that are growing globally in a model. I think in the early days of disease modeling decades ago, not considering what you guys are considering today, is take advantage of all the data. Right. And take advantage of negative and positive outcomes. I mean, a lot of people, negative, gone, move on to the next chapter. Every experiment, these expensive, well-controlled, well-done experiments are an opportunity for new innovation. Right. And every one of them contributes to disease modeling. Now, I appreciate that in the industry, we keep a lot of this information to ourselves, but figuring out a way to look at collaborative ways to integrate this data, publish the data, will advance how we think about diseases. So I applaud your efforts. I know it's not going to be trivial, right, but I applaud the idea of building disease models with real-world data, human metadata, and outcome data, hopefully coupled with quality multi-omics data on the immune system, because the immune system's getting implicated in everything these days. The more we understand about it, the more we're going to be able to look at opportunities across even broader therapeutic opportunities, particularly if we get into the CNS and other areas where many of these systems also play, which is happening.
Well, I agree, and I think if just to put the modeling component on this and kind of take a a step back into general machine learning, right? I mean, basically, in many ways, if I kind of take what you've been saying on how the industry and drug development is doing, we're missing the tools needed to learn and improve, right? So you need data, you need processes where there's a feedback loop that looks at a failure and then studies and improves the next time, and it requires data to do it, right? And you need many such things in many different settings, the breadth, and all of those are basically just now coming into play. So maybe that once the data is there and the processes and bringing the computational modeling into the beginning of the trial, my hope is that we will not just see the lucky step function, but also the more.
No, I think I'm aligned with this, and I think as many have talked about, data is not equal knowledge. So how do you take this data and not just look at the outcome analysis in your very elaborate multi-box scenario of where drugs will work or not work, but actually look at the why. And the why is hard. It's not been an area where people have investigated this, but this actually could be the opportunity for private-public partnerships to begin to look at that. And particularly with the kinds of tools that CytoReason has built, the opportunity to start taking modeling to the next level. Because although many companies, including my former company, were using Cray computers and many other technologies for decades and decades and decades and understand the chemistry of things, this crystal structures and all these, we're still not good at modeling biology.
Yeah, the biology's been left behind. I'd like to say the chemists run faster than the biologists.
And I think this is a great opportunity, probably with some focus on an important set of targets to show what machine learning, other algorithms can do. Because today this is still a black box to many groups. But teaching your models with real world data, outcome data, and then modeling, and again, getting some brave souls who want to pioneer the space with you to do this, I think is the way to change minds.
No, I think that's a wonderful way maybe to transition into the end of our journey here together on the podcast, and maybe just ask as you kind of been looking this far and all the kind of companies you've been advising at this point, what do you see as a commonality for how AI kind of changes the industry? Like, what makes you choose a successful company you're working with?
Yeah. Well, as we said before, we have to incorporate all the knowledge possible. And if someone would do the hard work, the legwork to build disease models, I think this is going to make people accepting this a lot easier. As you know very well, and I know from my brief experience at the Allen, finding the people that can build these models is not trivial.
No, not at all.
And so, having had a systems biology center in Singapore in the past, and then the Allen Institute, you really have to make a commitment to this space. And
commitment to modeling rather than developing the drug.
And taking, because almost all of these experts are involved in incredibly important design of trials and powering and all these interesting models, but they're not models of biology.
Yes.
So I think we have to look at the next couple of decades saying, no, it's biology's turn.
Yeah. I think that's a wonderful way to end our podcast. Tom, thank you so much. It's been really an educational experience and always fun to have you.
Well, thank you so much for inviting me. Enjoyed our discussion.